Hello everyone, my name is Christina Kramer and I'm a Global Product Manager for cell separation here at Miltenyi Biotec in Germany. Thank you very much for joining our live webinar about the isolation of PBMCs and cell subsets directly from blood products. The focus of today's webinar will be on how to use MAX technology to efficiently isolate functional primary cells without having to go through the tedious process of density gradient centrifugation. While we will be talking about the isolation of specific immune cell populations, we will definitely focus a bit more on our innovative and also efficient new approaches for obtaining clean PBMCs without having to do FICO. And therefore, we will start with a quick company introduction and we'll then have a brief look at MAX cell separation to discover where you can get primary cells from and also how MAX technology works. Next, I will show you how you can skip density gradients by using our new solutions for generating PBMCs or isolation of immune cells. In addition, we will have a closer look at how to automate these processes with our cell separation instruments in order to speed up the isolation and to make sure that you can work on other tasks in the meantime. In the end, there will be about 10 to 15 minutes for questions and answers. So let's get started. At Miltenyi Biotec, our slogan is to empower discovery and advance therapy. And as you can see described here, our mission clearly is to advance scientific research and medicine by providing solutions for cell and gene therapy, as well as biomedical research. Therefore, on one hand, we have a portfolio for research use only products, while on the other hand, we also provide GMP and clinical grade products. Miltenyi Biotec was founded in 1989 by Stefan Miltenyi. The basis of the MAX cell isolation is the column technology, which was developed as part of his diploma thesis and patented in 1990. Already in these beginning stages, Stefan Miltenyi had a pioneering spirit and saw the potential for MAX technology to become a revolutionizer in the field of gene therapy. The developments went on for over 20 years, including setting up a GMP manufacturing site in Tetero in Germany and also getting FDA approval for the Multimax CD34 reagent system. And so what started out as just one man's diploma thesis has since then grown into a global company of more than 3,000 employees worldwide, with almost a quarter of all employees working in R&D, engineering, and clinical development, and more than 20,000 scientific publications citing MAX technology. Annually, more than 2,500 patients receive cell products manufactured with the Miltenyi Biotec's technologies. So what is MAX technology, the technology that really everything started with, actually about? MAX technology is based on three components: MAX microbeads, so the reagents, MAX columns, and the MAX separators, so the magnets. And these three components together enable the magnetic separation of cell populations based on surface antigens. MAX technology really is so simple, and this is what makes it so genius. So let me briefly show you how it works. Only 3 steps are needed. First, we have the magnetic labeling, where an antibody conjugated to a tiny microbead binds to a receptor or protein on the cell surface. Step number 2 is the magnetic separation, where the cells are passed through a column that is positioned within a magnet. The cells that are labeled with the microbeads are retained in the column, while everything else is washed through in a very gentle way. The flow-through at this step is called the negative fraction, containing the cells without the ligand for the antibody or the antibodies that were used. And in the elution step, so step number 3, the column is removed from the magnet, which enables the magnetically labeled cells, so the positive fraction, to be washed out. All of this can be done either manually with a plunger to gently push the cells out, or it can also be automated, for example, by using a vacuum to pull the cells out, or by increasing the flow speed of the buffer in the system. All three methods are very gentle for the cells and separate the cells into the negative and the positive fraction with very high purity and viability. So the magnetic separators in a column-based and column-free system have a comparable magnetic force. The difference between the systems is simply the column that is filled with little iron spheres. Each iron sphere passes the magnetic force on to the next one over a very small distance, thereby amplifying the magnetic force by 10,000-fold and ensuring that the magnetic field is constant throughout the column. Thereby, minimal labeling of the cells with the microbeads is already sufficient to retain target cells in the column. The iron spheres have a cell-friendly coating, and moreover, the cells can run freely through the column and can be washed out thoroughly to remove cell debris and contaminants. All this together contributes to the fact that Max columns allow for cell separation with minimal effects on the cells, and this is exactly the reason why we use a column-based system. If you would like to learn a little bit more about the technology itself, its benefits, and if you would also like to see some data on this, feel free to have a look at the webinar recording Select the Best with Max Technology on our website. The link will also be included in the recording of this webinar, or if you just search for cell separation in the video library on our website, I'm sure you'll also find it. So, starting with Max Technology 30 years ago, Miltenyi's product portfolio has grown significantly, and the translational approach from benchtop to bedside is reflected in all our products. We provide the same products in research grade as well as GMP and clinical quality to ensure smooth transition into the clinic. We provide the entire workflow of products for cell-based work, from tissue dissociation and homogenization to cell culture, cell isolation, and also including activation and expansion, all the way to cell analysis and imaging. So, as you might have noticed by now, at Miltenyi Biotec, everything we do is centered on cells. But where do you usually get primary cells from? How do you work with them? And also, how do you prepare them to be used for downstream assays? Let's have a look at obtaining primary cells and all the steps involved, from your starting material to sample preparation, followed by magnetic cell isolation and the downstream applications. Every step to your target cells, including the sample preparation and cell separation, are actually crucial preparation procedures to ensure good downstream applications. Only if you prepare your cells properly, use the ideal separation methods and strategy, you will be able to obtain good data and results that you can really trust. While primary cells can of course also be obtained from tissues, most human primary cells come from blood products. And there is a lot of flexibility in this area depending on the number of cells you need, from whole blood or bone marrow with limited cell numbers to buffy coats and Leukopaks, which are definitely larger in size and contain a lot of white blood cells. So let's have a closer look at these blood products. When we explore the main sources for PBMCs, of course, whole blood is probably the best known starting material. In human peripheral blood, we find about 2 million PBMCs per milliliter. The volume is of course variable and the blood can be provided in different anticoagulants. The HCT, so the hematocrit, is rather high with about 44%. While the PBMC concentration in whole blood is rather low, buffy coats contain about 5 times 10 to the 8 PBMCs in a volume between 30 to 80 milliliters, but usually what we find on average is about 50 milliliters. A buffy coat is the waste product generated from a whole blood donation, and the HCT value varies between 4 to 10%. The same HCT can also be found in LRSCs, so the leukocyte reduction system chambers, are also called buffy cones or Trima cones, depending on the country that you're in. These cones contain on average 1 billion PBMCs in a rather small volume of only 10 to 15 milliliters. Similar to buffy coats, also LRSCs are a waste product, but in this case, the waste product of a platelet apheresis. The highest PBMC concentration can be found in Leukopaks, which contain up to or sometimes even more than 10 billion PBMCs in a full pack. The volume can range between 80 to 200 milliliters and sometimes go even up to 300 milliliters, while the HCT is very low, with usually less than only 3%. As you can see, there are clearly a lot of differences between these starting materials, and it is very important to know exactly which material you are working with in order to use the right cell isolation product and protocol. We have now discussed the different blood products in detail. Next, the sample preparation step is traditionally density gradient centrifugation to get PBMCs. We will talk about this step in more detail in just a minute. We have seen these 3 steps already previously, and depending on your project, you have different options to select from. Would you rather go for an easy and fast positive isolation, or do you need your epitopes free for a second sort, so you prefer untouched isolation? Or do you have many samples and would prefer to separate them in parallel compared to one after the other? Would you rather freeze aliquots of your PBMCs or isolate cells directly? All these factors are important in deciding which strategy is right for you at this step. Once you have your population of interest, you can move on to the downstream application. Whether it's an additional separation step of subpopulations, for example, Tregs, or flow cytometry such as immunophenotyping, we can recommend the kits that will make these steps easier. Or will you culture cells in vitro assays or run genomics or proteomics applications? Whether you are interested in maximum recovery, such as for cell culture assays, or high purity for reliable genomics or proteomics studies, we can help you with the strategy to get there. Not surprisingly, we will be focusing on the PBMC generation step and cell isolations first. As a summary of what we have learned so far, in traditional approaches, we go from blood product to PBMCs via density gradient centrifugation to get target cells in 2 separate steps. For this, we have the Max microbeads for positive isolation and isolation kits for untouched isolation. In this process, you have to go from sample to target cell via Ficoll. And at this point, I would like to do a quick experiment with you. Picture a buffy coat and 50 milliliters of whole blood. Both products just arrived in your lab. It is 3 p.m. on a beautiful sunny afternoon. The sun is shining and the temperatures outside are just perfect, not too warm and not too cold. You get a text message from a friend to go out and enjoy the beach or the lake or go for a small hike. However, you cannot join your friend because you will have to spend hours in the lab to isolate PBMCs from the buffy coat and the whole blood that you have just received. Let's be honest, this is not a very motivating or inspiring thought, right? So I'm sure I don't have to tell you what FICO looks like. I'm not going to go into detail here. There are many steps involved. You have to centrifuge, you have to wait for the centrifuge to stop, you have to count the cells, and it takes a lot of time and is prone to human error and user variation. And also, it will definitely not allow you to join your friend and enjoy the good weather outside. So on top of that, there are actually some more downsides to density gradient centrifugation, starting from the contaminations of red blood cells, platelets, and granulocytes in your PBMCs. Also, there are several studies that show the toxicity of Ficoll to white blood cells. I'm pretty sure that everyone who has done Ficoll knows that it is a laborious process that takes a lot of time. And also, it is highly dependent on the skills of the person who does it. And even if you have quite some experience, you may still get a low recovery of cells sometimes. Therefore, it is prone to intra and also interoperator variation in the harvesting of the interface. In addition, due to all the centrifugation and washing steps, the process cannot be automated, and it is necessary to stay in the lab. The good news is we have a solution to solve all these issues. And these solutions will also allow you to join your friend and enjoy the sun outside at the same time. So Miltenyi Biotec's Ficofree solutions allow you to isolate cells directly from blood products without density gradients, without cell counting, and in only a few simple steps. You can completely skip the density gradient centrifugation, and the protocols are very straightforward and easy. And this will give you the full flexibility as well in terms of isolation strategies. At the same time, these are the fastest protocols available for isolation of PBMCs and immune cells directly from the different blood products. And there is only very little hands-on. So this means user variation can be reduced significantly due to the automated options that you can choose from. So why is this important, especially right now? Under the current working conditions, you may be facing new challenges, unexpected new challenges. And I would like to briefly give some examples how our new kits can help you solve these worries. Due to the ease of use of the kits, so only one person is needed to perform the separation, as well as the fast and easy processing, social distancing guidelines can be followed at all time, even when the blood comes in late due to shipping difficulties. In addition, even when the time in the lab is limited, since in many cases people are working in shifts, the fast protocols and great performance nevertheless ensure to get the experiments done in time. We will hear a little bit more about automation later, but as you may already know, cell separation instruments ensure user safety by reducing the exposure of the operator to potentially infectious material. It also helps to focus your time and energy, and thereby reduce errors and the need of repeating an experiment if something goes wrong. For us, and I'm pretty sure this is also true for you, every cell counts. Therefore, of course, automated separations will also improve standardization and reproducibility. Having this said, we can now fill in the other option to Ficoll, the Ficofree solutions. They allow you to go directly from the sample to target cells without a PBMC step in between. Or you can also isolate PBMCs directly from the blood products without density gradients. You can then either use the PBMCs directly or take max microbeads or isolation kits to further isolate subpopulations from fresh or frozen PBMCs. Now, I have already mentioned so many benefits of isolating the PBMCs directly from the blood instead of doing a density gradient centrifugation. So let's have a closer look at how this new cell isolation method works to obtain pure PBMCs. Depending on your starting material, we have specific isolation kits available for the untouched isolation of PBMCs from peripheral blood, buffy codes, LRSCs, and Leukopaks. The workflow is almost identical. Therefore, let's have a look at the protocol in general. First, we sediment the erythrocytes using a dedicated sedimentation kit. Including the pipetting, this takes about 5 minutes. Afterwards, we take the supernatant and centrifuge for 5 minutes. After resuspending the cell pellet, we can directly continue with the labeling of the cells with microbeads by adding the reagents and incubate for 10 minutes. Next, the magnetic separation will be done on our cell separation instruments, which takes between 5 to 20 minutes, depending on your starting material and the volumes that you work with. For example, of course, it will take less time to process 10 milliliters of whole blood than 500 milliliters. When doing the separation on the Automax Pro Separator, which you can see in the picture here, already the incubation step can be automated. Of course, followed by the fully automated separation. In summary, you only spend 25 to 40 minutes to obtain untouched pure PBMCs from your blood sample. We can now take this one step further and compare the total time it takes from your sample to PBMCs. We have seen how long Ficoll takes, no matter what your starting material is. Of course, it also depends a little bit on the volumes, the dilution factor you work with, and therefore the number of tubes you need to handle. But just to have an example, it definitely takes more than 1 hour or even up to 1.5 or 2 hours. And this is what you can see here in the purple bar. When we compare this now to our new PBMC isolation kits, we immediately see that the protocol is much faster, independent of the starting material. When working with whole blood, buffy coat, and buffy cones, only 10 minutes of hands-on time are required for the preparation and sedimentation step. So this means sedimenting the RBCs, taking up the supernatant, and spinning for 5 minutes. Afterwards, the process, including the labeling and separation, runs fully automated. For the Leukopaks, the initial sedimentation of RBCs and the centrifugation of the supernatant can be skipped, and we can start immediately with the labeling, followed by the magnetic separation. We can take this now one step further and ask, how much time would it then take if we get, for example, 2 buffy coats and want to process both of them on the same day? Let's find out. We would start working with the first buffy coat, do the sedimentation, and then start the labeling and fully automated incubation and isolation on the Automax separator. In the meantime, while the instrument is running, we can take the second buffy coat, also do the sedimentation of the RBCs, and then start the labeling and incubation. The PBMCs of the first buffy coat will be available after 40 minutes in total. So once this is the case, we can then start the separation process of the second buffy coat on the Automax. If we sum up the time, instead of 80 minutes, so 40 plus 40, it will therefore only take 55 minutes to obtain PBMCs from 2 buffy coats using this staggering option. So far, so good. But I'm pretty sure you're also interested in having a look at performance data. Here we go. When we compare the cell composition of the PBMCs we obtain after a regular Ficoll and our new PBMC isolation protocol, we can directly see in this dataset that there is no variation in the cell composition. The starting material in this case was whole blood. And the only difference that you may notice is that the granulocytes are actually not present anymore in your target cell fraction after using the new PBMC isolation kit. And this is actually great news, as PBMCs consist of lymphocytes, monocytes, whereas erythrocytes and platelets, as well as granulocytes, do not belong to PBMCs. If we have a closer look at this slide, I would like to show 2 things when we compare the performance between Ficoll and the new PBMC isolation kit. First, there is no granulocyte or erythrocyte contamination in the PBMCs when using the new PBMC isolation kit. So if you are interested in very pure and clean PBMCs, this new method is the perfect fit for you. Second, independent of the donor that you get, the reproducibility is given. Since the method is based on max microbeads, donor-to-donor differences do not impact the quality of the separation or PBMCs. And as you can see in the picture, when using Ficoll, differences between donors do have a more significant impact on the performance. This data was generated with whole blood as starting material, but let's have a look at buffy coats as well to use another example. As you can see here again, the composition of the PBMCs after using Ficoll and the PBMC isolation kit is comparable. However, with the new PBMC isolation kit, the obtained PBMCs are much cleaner since there is no RBC or granulocyte contamination. For us as scientists, numbers and data is definitely an important argument. But sometimes a picture is worth 1,000 words. These pictures are of course not professional photos, but simple pictures we took after isolating PBMCs with customers that are already using the new kit. In all the pictures, you can see the results of the PBMC isolation. Only in the first picture on the left side, you may notice the reddish color in the right tube. In that case, we actually did a direct comparison from the same sample using Ficoll versus the PBMC isolation kit to generate PBMCs. And as you can see, when using Ficoll, the red coloration demonstrates that there are still a lot of RBCs in the target cell fraction, while the PBMCs in the other tube, so the tube on the left side, are completely depleted of erythrocytes after using the PBMC isolation kit. So at this point, we have finalized the section on isolation of PBMCs. Let's move on to the separation of specific immune cell subpopulations directly from blood. We have seen this overview before and will now focus on the straight from portfolio to isolate target cells directly from blood products. Again, also in this case, the protocol is very similar, independent of the specific starting material that you work with. No matter if we're talking about whole blood, buffy coats, LRSCs or Leukopaks, first, you fill the blood sample into a collection tube. Next, straight from microbeads are added and you incubate for 15 minutes in the fridge. Then the magnetic separation is done on the Multimax separator, which you can see on this picture. Or in the case of whole blood, you can also use the Automax Pro Separator. In both cases, it just takes 30 minutes from the blood sample to holding your target cells in your hands. Straight from is based on a positive isolation approach from the starting material. And this actually provides a very attractive advantage. Positive selection enables multiple consecutive separations from the same sample by using the negative fraction for another round of separation. Here we have an example. First, you isolate CD8 T cells and then take the negative fraction for isolation of CD4 T cells. And you could even take this further onto another level and take again the negative fraction to then isolate another cell subpopulation. Therefore, if you are interested in several target cell populations from the same sample, this may be the ideal product for your experiments. Cell isolations with straight from are possible from all the different starting materials, as I had already mentioned. And as we had seen, the protocol is quite simple, fast, and straightforward, without density gradient centrifugation, cell counting, or any washing step after the labeling. And here you can see just one example for isolation of CD4 positive cells with the straight from Leukopak CD4 microbead kit, before and after the separation. To support that with some numbers, on the right side you will find some purity data generated from buffy codes for a variety of markers. The purity in all cases is 95% or higher. And it is actually just a small fraction of the markers we have available. So make sure to check out our website and the link below the table to find the list of markers that are available. And we regularly release additional new products within this portfolio. Actually, the newest addition are the straight from buffy code release microbeads, which provide bead and label-free target cells. Make sure to check them out on our website. In addition to just looking at purity and recovery, of course, we also test activation, cytokine release, proliferation, viability, and just many more properties of the isolated target cells. Actually, I could fill hours showing you all this data. So just picked one dataset about the activation marker analysis to give you some quick insights. Here we isolated CD4, CD8, or CD4, CD8 T cells with the straight from buffy code microbeads. The cells were either stimulated with Tcell transact or were kept without stimulation. The activation markers CD25 and CD69 were then analyzed. As you can clearly see in all cases, there is no activation of cells after separation with straight from, while cells could be successfully activated upon stimulation. If you would like to see the straight from kits in action, feel free to have a look at this video on our website. Moving on, I would like to spend just a few minutes on one of our application highlights with the Straight From Kits, our research CAR T-cell engineering workflow. For the CAR T-cell generation, you will always start off with a blood product, like all the blood products we have discussed earlier, to isolate the T-cells. Next, the isolated T-cell population will be activated, transduced, expanded, and the CAR T-cell functionality will be analyzed. So for the first step, the enrichment of the T-cells from blood products, we basically have two options. Traditionally, the first option is to generate PBMCs and then isolate T-cells from these PBMCs. Here you can use the CD4 or CD8 microbeads for positive isolation, or you could also use the T-cell isolation kit for negative isolation. As you already know by now, we also have a nice alternative available, isolating T-cells directly from the blood product with Straight From. Specifically for the CAR T-cell engineering, we did a side-by-side comparison of Ficoll plus Max microbeads and the direct Straight From microbead approach. We checked for T-cell enrichment, T-cell activation, the transduction efficiency, and the target cell killing. And we observed, as expected, comparable results. But, of course, the cell separation step was done much faster using the Straight From approach. This means that the isolation approach you choose here will have absolutely no negative effect on your downstream results, but will allow you to be much more efficient. If you would like to learn more about CAR T-cell research, please feel free to check out this webinar recording, our latest solutions to improve your CAR T-cell research, on our Miltenyi Biotec website, given by my colleague Saskia Rösch this year. So we have now covered the generation of untouched PBMCs without Ficoll by using our new PBMC isolation kits, as well as the positive isolation of target cells directly from blood products using Straight From microbeads. For specific functional assays, like for example antibody-dependent cellular cytotoxicity assays, or for example for isolation of specific cell populations, you may however want to isolate untouched target cells. And you may also want to skip the PBMC step here as well. We are currently developing a broad spectrum of kits to do exactly that. Isolate untouched immune cells from blood products without density gradients, speeding up your processes while providing excellent cell yield and purity. So, does this sound exactly like what you've been looking for? And are you curious to learn a little bit more or to even be a beta tester? If so, and if you want to get a sneak peek and would like to work with our fully new products before their release, either reach out to your local sales rep or just send an email to me directly. With this, we have now covered the reagent perspective of this presentation. And as mentioned in the beginning, I would like to just spend a few minutes on the automation of cell separation processes. So, why do we actually need automation? What is the huge advantage? As we have already said a little earlier, automation reduces hands-on time and frees you to work on other tasks in the meantime that cannot be automated. It also minimizes variation between different users in the lab, or even good and bad days of individual scientists. And of course, it minimizes costly human errors. With standard protocols and minimum user variation, you can be sure that the effects that you are seeing are due to your assay rather than artifacts. In short, automation therefore reduces redundant experiments and allows you to focus your time and energy where it really matters. So very shortly, I would like to introduce the Miltenyi Biotec automated solutions for magnetic cell separation. And actually, you may already recognize them because I had mentioned them a little earlier already when we looked at the new protocols. We will start with the Automax Pro Separator, the easy-to-use benchtop instrument for every lab. With fully automated sequential separation of 1 to 6 samples, the Automax provides easy access to automation without high investment cost or a lot of training. You can mix many different starting materials and samples within one run. Press the Run button and the instrument will label and separate the cells for you. The columns are reusable, so maintenance is very easy and the column cost can be reduced. The instrument is actually the perfect match for the isolation of PBMCs from whole blood and buffy coats. But of course, also target cells can be isolated from these PBMCs with the Automax. If you are interested in parallel cell isolation, the Multimax family is perfect for you. 24 magnets allow you to process 1 to 24 samples in just one run, or you can also split large samples between the multiple columns. This is achieved by using LS, LD or whole blood columns, or you can also use the multi-24 column block for easy handling of 24 samples or very large sample volumes, like for example an entire Leukopak. The Multimax Cell 24 is a semi-automated instrument and a good fit for many labs that value easy handling but don't mind the manual pipetting. However, if you want full automation and maximum throughput, you will just love the Multimax X, which integrates a Multimax into a liquid handler. So this instrument is actually the perfect fit for demanding high throughput cell processing labs. Samples are labeled and isolated fully automatically, both for the PBMC generation or cell subpopulations from all blood products. All programs are tailored to match your needs, so we don't leave the lab until you are satisfied with the protocol and the results. In summary, therefore, the Automax is really great for PBMC isolation from whole blood and buffy coat with full automation. Because the Automax can process whole blood without Ficoll for isolation of cell subsets, it's also great for patient samples or small donor samples. In addition, it is also the instrument of choice for the low throughput isolation of cell subpopulations from PBMC aliquots. On the other hand, the Multimax family are the high throughput variants for fast parallel processing, for example for PBMC isolation from Leukopaks or isolation of cell subpopulations from large volume blood products. Depending on the level of automation needed, you can choose between the semi-automated Multimax Cell 24 and the fully automated Multimax X. As we have learned, no matter what your starting material is, how often you get it, how many samples you want to process in parallel, we have the instrument that will exactly fit your needs. So we are coming to the end of my presentation, and there are just a few things I want you to keep in mind when thinking about density gradient-free solutions for cell isolation with Max technology. It is no secret that the success of any experiment depends on the quality of the starting material. So remember that the right cell preparation and isolation steps are crucial in the beginning of any workflow to ensure reliable assay results. Automating these processes with the help of our cell separation instruments, combined with Ficofree protocols, allows you to be more efficient while ensuring high reproducibility and standardization. Our new methods enable you to isolate your target cells or PBMCs directly from blood samples with high speed, great performance, and easy protocols. Of course, our disclaimer cannot be missing in any presentation. But to finalize, I hope I was able to give you some insights into our world of density gradient-free cell separation. And feel free to either reach out to your local sales rep or send an email to me directly if you're interested in more information, if you would like to be a beta tester, or if you would like to get a demo. Thank you very much for joining.